Okay. Oh, I see what you mean. Move it. Okay. Okay. Welcome to day three's workshop for cancer genomics. Just before today's class begins, there's a QR code about the survey of this workshop. Just to let you know, this is the first time I teach this workshop, so your suggestion is an invaluable resource for me to further improve the quality for this workshop. So please take a moment to scan this QR code to fill this survey during or after this class, please. Okay. If everybody has scanned this QR code, we can finish this survey alongside or during the break. I just want to make sure before any of you leave, you scan this QR code. Okay. Okay, so everybody scan this. Let's start with today's workshop. And before that, let's take a brief overview of what we learned from yesterday. So yesterday, we mainly threw this R package called TCGA BioLink, and we set up for quite some time to install and set up our system environment. Some of you have asked me the questions, what are the source that these packages come from? They come from two sources. One source is called CRAN, which is the official source for those repository packages that are official data repository from our language. And this is we install those packages by this command line called install.packages. These are called the packages from the repository of the official R repository. And we installed this package Bio Manager, which is a package help us to install additional packages from another source, which is called a bioconductor, which they store the multiple very useful packages for bioinformaticians for specifically for genomics or proteomics that related to biology. So that's what we use for the additional method. We call the install function from this Bio Manager package, and we install the rest of those package from this Bio Manager repository source. Hope that answer your questions. And after we install those packages, we use two cases examples to show you all how to query and retrieve and load those data sets into the our system. We use 5 cases or several cases of somatic mutation data sets to plot some basic analysis, such as the gene mutation rate or translation versus transversion rate, etc. And then we mine all of the transcriptional data sets from the TCGA read cohorts and then do the DSeq analysis. And then after we successfully run the DSeq program, we also do some actually gene data format sorting to merge the data frame that contains the gene names with gene ID with our results data frame from the DSeq results. And then after we transfer the data, the gene ID into the actual gene name, we use the enhanced volcano plot to plot the volcano plot to show the gene name that are significantly different, so expressed in our cancer versus our normal cohort. Okay. So, excuse me, I think I should. Yeah, okay. So today, I will also use 2 examples to show you all how to perform some downstream analysis by the data sets that we retrieved from yesterday. We are also going to use the same data sets from the transcriptional data sets we downloaded yesterday. The first example, and will be used this DSeq pipeline again with our transcriptional data sets. We will be leveraging some normalized data sets from DSeq intermediate output, and then build a so-called UMAP projection, or uniform manifold approximation and projection. This is algorithm belongs to a dimensionality reduction algorithm, which is I personally call that pseudo single cell analysis, because this method is constantly used in the single cell RNA-seq analysis. And the second case, we will retrieve and knit the format for the clinical data sets from the TCGA read projects. And then we will combine that with the transcriptome data analysis that performed by the DSeq. And then we will pick a selected oncogene that we're interested in, and then differ our cancer cohort into two groups based on the transcriptional intensity of our selected oncogene or the gene of our interest. So if one group, for example, we're going to use a oncogene like CDKN1A, for example, if their one group's expression level is high for that gene, we categorize it into group 1. If the other group will be lower expression level for that oncogene, and then we'll plot a Kaplan-Meier curve to compare the survival rate between those two groups. So that will reflect whether that oncogene will correlate with the patient's survival. Does that make sense? Do you have any question? Okay. So let's go into the first example. So first example will be performing the UMAP analysis, which belongs to this dimensionality reduction algorithm. So some of you may already heard this term UMAP, or this full name uniform manifold approximation and projection. And the principle belongs to their dimensionality reduction algorithm. Is I can give you some of an example like this. So for example, we have a cohort of data sets that we only have two variables about the data sets. One variable is height, and the other one is weight. So we can plot a dot plot with the weight on the x-axis and height on the y-axis. And then this dot plot will be very easily to distinguish the two groups of our data sets based on the similarity on their on the plot. On this two-dimensional figures, it'll be very easy to present the clusters. So for example, we have a third variable called age. So we can still cluster our samples on the three-dimensional figures. But the issue becomes when we have more variables than three, we can plot four-dimensional data sets in the three-dimensional figures, especially in our case, what if we have 20,000 gene expression levels as variables? We definitely cannot plot 20,000 dimensions on this figure. So this has become necessary for when we use this so-called dimensionality reduction algorithm. They're basically doing is calculate a projection from a higher dimensional data structure on the lower dimensional figure. So it's similar, it's a metaphor like what we've seen here on the two-dimensional graph is actually the shadow from higher dimensions. So for those dimensionality reduction technology, there are three that are commonly used in our genomic field, which is principal component analysis, or PCA for short. Another one is called the t-distributed stochastic neighbor embedding, which is a very tedious term, short for t-SNE. And the third one is UMAP. The PCA algorithm, which is their mathematical principle, is relatively old. probably their mathematical function is over 100 years old, which is usually used to quality control of our data set. It reflects whether our data set has a statistical difference from another group, or are there batch effects from different batch of experiments. TSNE and UMAP, on the other hand, use a more advanced machine learning algorithm that is published in recent decade. I think it's only 5 or 6 years old of their algorithm. Maybe I'm wrong. Maybe there are. So maybe they're about 2015 or 2016 year when their algorithm is first being published. Those two algorithms are constantly used in single cell data analysis for identifying different clusters of their single cells. So what we are going to do is we're going to use their single cell data analysis method on our bulk RNA-seq data sets. I think this is a pretty interesting methodology because recent years, more and more papers and researchers have starting to use this UMAP function on bulk RNA-seq analysis. What I'm showing as the evidence is a paper on the left from the Cell Report demonstrates the quality on UMAP algorithm is better than using the traditional TCGA method for clustering bulk cells, bulk RNA data sets. And the paper on the right shows a case that researchers are starting to use this UMAP to cluster bulk RNA-seq analysis. So when I was doing my research, I tried both methods on the TCGA READ project, compare this PCA algorithm versus the UMAP algorithm on clustering different cells. For this dimensionality reduction figures, each dot on this scatter plot represents a transcriptome from a single patient. The physical distance from dots indicates the transcriptome similarity between different patients. As you can see here on both left and right figure, the cancer patients or the cancer transcriptomes is colored in red, which is more dispersed, but they're clustered into a unique cluster compared to most of the control healthy normal samples. And compared to these two data sets, even they only have a relatively small sized data sets, around 100 and 200, we can slightly see the trend that it seems like a UMAP have more unified cluster compared to the PCA a little bit. This becomes more and more obvious if you use your own large cohorts of data sets, such as if you increase the dot by 1,000, probably the difference will be more obvious. So, any questions? Okay. So I'm not going into the mathematical details about those methods, but I pasted a link here on the YouTube link that vividly displays or using a metaphor to show how these three different methods are being calculated to generate the projection for the UMAP and the TSNE. If you're interested in the mathematical principle, feel free to check this YouTube tutorial. So in our workshop, we are mainly going to learn how to use that function on our bulk RNA-seq analysis. So let's plotting the UMAP on our code. Okay. So this is from yesterday's code. And let me open a new R code for day 3. And the first step for today's code is we need to load the necessary libraries for today. Load our libraries. Since we have already installed all the necessary libraries for yesterday, today the libraries we will need is TCGA biolinks. and then summarized experiment, library tidyverse, library seq2. We don't need math tools package anymore because we're not analyzing the mutational data sets for today. And then we will need a package called UMAP. And then the package called survival. Finally, the package surfminer. Those are the necessary libraries for today's workshop. So I hope everyone has successfully loaded those libraries, right? If we successfully load those libraries without error, the next step will be set up our working directory. I recommend you all to set up the same working directory from yesterday so that we don't need to download the datasets again. We can just load the datasets to the path that where we downloaded from yesterday. So I'm going to use this command line to set up my working directory. And then I'll go to the terminal to the location of my project. So here's my working directory, and you can change it to the path of your directory where you download data from yesterday. And after we set up working directory, we get a double check to make sure it's properly set up by use this get working directory command line. Okay, it prints out the directory is where we set up. And then let's do the same query from yesterday transcription datasets. So I am going to steal some of my code from yesterday where we built the same criteria to retrieve the transcriptional datasets. The data is in day 2's part when we build this RNA query with this. When we first made this RNA query is equals to GDC query or project filter, data category filter, access, and a sample type filter. I am going to copy this one into our day 3 script here and run it. And we can always give it a check of our results if they are the correct format by using the get results function to check our RNA query if it's the formats that we want, if those are the actual files we downloaded from yesterday. We just nominated this into a new object, output RNA underline query, make it equals to this function. And we can view our output to make sure those are the files from yesterday's setup. So those are the TSV formats with open access of transcriptome profiling data categories files. And the file number is 176. Everything seems to be normal. So today we don't need to download it again because it's already in the directory. So we can just load it by using the GDC prepare. And then the input will be RNA query. And then let's nominate it into the object called RNA. Make it equals to this. And as you all see here, it automatically recognized the location where I downloaded and loaded properly. So this only successful without generating any error, only if we set exactly the same filter criteria with our RNA query. If we change a file number or something, it pops out error, some of the files are missing or something. Prepare your data into the R system, into the R Studio. It's saved in your hard drive, but you need to prepare it into the R when you can actually call it. Yeah. Yes. Yeah. Yes. RNA query object, it's kind of like the data sets we created from day one's website. It's like a data set, but the query is actually in the repository when we call it. I don't know if that metaphor makes sense. Or the query is when we built the setups, we built the different filters. We filter such as we filter only on transcriptomes, we filter this, we filter that. It's like in the the exploration page on the website. However, the prepare is like a little bit like a repository page where actual files are being loaded. Yeah, that makes sense. Oh.
Yeah. And we can use the GDC prepare after we have all GDC download.
Yes. Yes. This function prepare can only be load the files stored in your personal hard drives. Yes.
You're saying you can make a folder, like for example, for like the GDC data, then just make the working directory the same folder so that it can easily access those files.
Yes, so that's why we set up the same working directory as yesterday. Yes. Hope everybody have successfully load this one, right? Okay. Now let's transfer or let's extract the unstranded column from the raw data, which is the actual gene counts, by using the same command line or the code we did yesterday. We nominated a new matrix called RNA matrix. Make it equals to our assay function from our RNA object, and then we extract the column called unstranded. Okay. Oh, yes. Assay. Oh, wait, how to spell? Okay. Okay. Now we can check the matrix by using command line head. to show a few lines of our input files. Okay. It print out the first 5 lines of our assay matrix, which each column is our patient case in TCGA barcode, and each row is the genes in ensemble ID with the raw gene name, raw gene counts. So this is this matrix. So the matrix format is correct, and then we can load that object into the seq like we did it yesterday. So we create a DDS input, make it equals to our DE-seq data sets from matrix function. And then inside the function, count data equals to our RNA matrix. And then the code data equals to our code data of RNA object. And then the sample type for our condition in design equals to our sample type. So we successfully load those data sets into R, into the seq2 package. And then like yesterday, we filter the low counts based on a threshold that we are comfortable with. We are creating a standard called keep, make it to row sums or row median or whatever threshold that makes you feel comfortable. And then make it equals to the counts of our input. I'm going to keep the threshold as same as yesterday, make it to above or equals to 10. Okay. Now I am having an error. Counts, yes. I'm missing a spell. Okay. And then we can filter our input by keep the lines only it satisfied with this keep filter, which is above or equals to the row sum of 10. Okay. Now the filter is like 44,000 lines of genes, like the same of yesterday. So we can run the DE-seq program by DDS equals to DE-seq our input. This will be the same procedure like we did late yesterday. Yes. Yes. And that's yeah, this is a symbol like how you want to set up your compare groups, such as we set up our compared group condition by sample type. And you can actually change it to some other parameters, but it needs to be only have 2 variables because DE-seq only can take 2 groups for comparison. Yeah.
And how does these
You can actually create this into a new object, such as sometimes I've seen in some people they will create like a condition, make it equals to like condition 1 is from file 1 to file 50. It's something like that, and then the design will be equals to this condition. I hope everyone has finished the DC. Is everyone's computer still running? Finished? Okay, give me a thumb up if yeah, still running. Okay. Mhm.
So the count data we provide that is on a matrix.
Yes.
where we call the count data from one, and then where it does, like, is the sample type part of the whole data?
I think we can do this. It's in a function called, I think it's column names of our RNA object. Sorry, it's not this. It's in oh, give me a second. I think I can find that script. Um, it's in RNAs, and then yeah, it's in its RNA object. One of the a column in the RNA object is called a sample type, I guess. And if you can just call that sample type from this and print out each sample's sample type. And besides that, it has other names. All data. Yes. And then
the row names correspond to the column names of this RNA matrix, and so that's probably how it matches to the sample type column.
yes. This column name will print out the entire if we head into this DDS object, it's like their column data, it's also have multiple inputs. We typically use a specific function such as assays to call the data or the designs to call some specific things. But they have other functions to call specific informations. And I found this column in this sample type of our actually RNA column so that we can just use this column calling to directly, yes. It's for which column. Yeah, it's actually the column data in the S4 object. This is S4 object is actually they have a unique parameter called column data. And the column is another object independent of that column data. So have we finished this one? Okay. So if everybody's DE-seq program has finished running, if you remember yesterday, we just called the results to see actually the differential gene expression analysis. But today we will do something else. We will perform a function from this DE-seq package called variance stable transformation. Variance stable transformation by using a function called a VST. And then our input DDS and then blind equals to false. Let's nominate it as a new object called a VSD, and then do. You can consider this a VST transformation function as a way to normalize your raw data. In their manual, they have specific description of how this formula of this function. And this function is usually used to perform QC check by the PCA methods followed by this one. For example, we can check dimensionality reduction by the PCA. And in DSeq package, there's a built-in PCA plotting function by just type this plot PCA. And then our input will be VSD, and our group by int group is our sample type. And then and then we can generate this PCA plotting like we saw on the slides earlier. It's a condition set whether we want to ignore control versus in this case it's the sample type as a difference. There is a calculation whether we want to ignore the control group with the experimental group and then calculate them all. We just typically would say false. Let's say it's whether blind transformation with experimental design. In our experimental design, is our sample type, whether to blind or ignore that difference between cancer or normal group. Is there? It's typically when we do the QC, we don't know whether which is experimental group or condition. We want to calculate a variance stable transformation with no bias. If they cluster separately, we know that it's actually because of their data structure is different. Okay, so let's do a comparison between this PCA with UMAP. And after this variance stable transformation, we can leverage these values to not only plot PCA, but also plot the UMAP. So to plot UMAP, we need to extract the data format from the VSD object into a matrix. Let's call that VSD counts. Make it equals to our assay VSD. Now, if we take a look into the actual format of our VSD count, we've seen that the variables and variance stable transformation of the values for each case with each genes in row. So this is a kind of one kind of a normalized value. And then for UMAP projection, we need to transform the data structure from rows to column. So basically, we want each row is a patient case, and we only need 2 columns indicating the coordinates on X and Y axis. So that 2 coordinates is the projection from higher dimensions. So what we need to do is to use a function when we are calling the UMAP by calling a function called T, which is transform the rows to column to shift the data frame of our VSD counts. And then after that, we can use this object to actually calculate UMAP. And then let's make it nominated into a new object called UMAP, make it equals to this one. Another parameter we can play with or set up with is called this a neighbor. Or this is called nearest neighbors. It's basically when we do the iteration of this calculation, it set up how many neighbors can each dots have around them. And this one is kind of important because let me show you an example of the results if we choose different parameters. So if we choose the parameters with the smaller numbers, it basically reflects more finer scaled of our clustering than the large number, which is a more of a global view of this clustering. I think we can think of this as using different distance to view this data, whether we enlarge it by 500 times or we view it from 500 times distance away to take a look at the structure of the data. So both of the data reflects the actual transcriptomic difference from each other. It's just by how much of the difference you want to present them. So for example, if you have a larger data sets, you probably want to set the number a little bit up to show actually the clustering. Otherwise, it'll be too much details. You subclusters your samples too much. You want each group to cluster them together. You can play around with these numbers to check which one is the best presentation of your figure. So by default, I'm just going to use this for 15 and do the calculation. Yeah. I'm going to run this again. Run this again. Yes, oh yes, this is the issue. Thank you. Okay, now that's fine. Yes, it's just to finish this calculation. It calculated into a results. Okay. Now and this result is actually also a somewhat S4 object, and we need to extract the data sets from that object into an actual data frame or data format that we can actually see. So what we're going to do next is we create a new object called UMAP results, make it equals to our UMAP layout structure, and then we nominate this layout as a new data frame. So no S. We nominate this as a new data frame that a function as data frame of this object. So now when we take a look at the actual results of the data frame, we have two columns and the row names represents to each case's barcode. And then we have V1 and V2, which is a two-dimensional coordinates of this projection from actually 20,000 dimension of data sets. Yes, yes, that's how I did when we plotted. So to plot this UMAP plot, it'll be quite easy. We just use the ggplot with the scatter plot to show that. So we'll be use the function ggplot. And the data is our UMAP results. And then the AES, which is the coordinates, is x-axis is our V1, y-axis is our V2. And then we give it additional information by we want to plot the point for each of the value. And then we can nominate our x-axis label as UMAP1. And y-axis is a UMAP2. Okay, now we generated this UMAP before we label the cluster. So probably each of you are generating a different shape of this cluster as I did. This time my figure is different from the first time that I saw. So this happens because like it's an iteration of a calculation. If we want to generate a reproducible figure from every time it shows the same, before we calculate the UMAP, we need to set a specific C's as initial numbers for the machine learning algorithm to take input. So what I'm saying is like, it's okay that each of the results, the figure looks different. It's because like the machine learning algorithm take different initial numbers to train their data sets. So if we want to make every time of our analyzing stable before we calculate the UMAP by this function, we need to set a specific initial number so that to tell the system, this is the initial number as input before you train. Otherwise, the system will randomly pick a number to start training. So that's why each time of a UMAP calculation, each time you run this figure, probably generating a different shaped format. But actually, the distance between different groups should stay the same that reflect the actual data structure difference. So if you, for example, if you want to publish the data when you're doing the reviewing process, so the reviewer after to regenerate that figure, you probably want to make it stable so that before this UMAP calculation, you can do a function called stat C's, and then you just randomly give it a number. So the system, each time run this function, they'll use this as C's to run. It generates the same UMAP as always. Yes?
Or like pseudo.
For
pseudo random process.
Pseudo random processes. There's another function. Is that what you mean?
I think that's what you can use this in general to do pseudo, like not just for UMAP.
Yes.
So it has
Yes. Not just for UMAP. I'm just using this as example. I think. Yes. It's just the initial random data sets that computer will use to start training this function. You can whatever number you put as input, and then this is only become useful if you want your every time of your UMAP to look exactly the same every time. But I sometimes just let it randomly generating a different figures. It's fine. Let's, yeah. Yeah. Yes, yes, we manually define our X or Y axis label as the name. It's the V1, UMAP1 and UMAP2 is just mathematical numbers for the coordination. It's actually not biological meaning anything because it's just a projection. And since we don't give it the set C's at the beginning, so I assume the computer take a random number, your random number start training is different from mine on the background when it start running. So that's why the output figure shape looks different, but their clustering should be the same.
So V1 and V2.
Yes, this. Yes.
They don't represent the reduction dimension.
It's the shadow of from 20,000 dimension to the two-dimensional, and then it's the coordination of each of the dots. But actually, it doesn't reflect any transcriptional intensity for individual genes because it's a mathematical calculation too much. But the distance from dot to dot actually reflects the transcriptome differences between those two dots. Yeah. Yes, it's just helpful visualization. We can do a 3D. We can do a 3D. I personally haven't tried that, but probably this UMAP package have a specific parameter that you can use to generate. I'm not sure. I personally never tried it before. And you can always check their menus by using this question mark and then give it a function that you want to check. If they have specific descriptions, you will find the parameters that you can use to set up. For example, if they have a three-dimensional generating things, they are listed here. And you can also check their website for each of the function. They should have a pretty terrible display. Yeah. So a good thing about this we call that a pseudo single cell analysis compared to the real single cell analysis is the sample labeling step. For actually the single cell data analysis, to label different cells with different samples is actually quite a difficult task, and multiple researchers has different papers and research such as focused on marker genes or etc. to label their cell cluster. But our bulk-seq data sets is already recorded from the TCGA information, such as we deferred our samples by control versus cancer samples. We already know that information in our data frame. So to label that, we can just manipulate with actually the data ggplotting, to plot the data. We can actually add another parameter called color to further refine that. But before we do this step, we need to merge the information that we want to cluster with actually the UMAP results. As we all see here, the UMAP results, the row name is the barcode number, and we can just merge the barcode number with the clustering information we want to show. So I'm going to create a new object to merge that with the actual information by nominating it as UMAP merged. Make it equals to, I'm going to use a command line called cbind to bind our UMAP results with our variable varying stable transformation output called VSDs code data. This as a new function called label UMAP plus by By doing this, we combine the information from this VSD's code data with our UMAP results. And when we take a look at the UMAP results, besides the sample barcode and the two-dimensional coordination, we have additional information from that very stable transformation. And this have in total 113 different columns that we can give it a try for the things that we are interested in. So for the simplest one, we can cluster based on the sample type. So what we are going to do is using the same ggplot function. And this time the data is our UMAP merged. And then our X and Y axis stays the same, V1, V2, and then we can give it different labeling by a different color. Make the color, let me change it to another line. Make the color equals to, in this time, is our UMAP merged data frames sample type. And then the rest of them stays the same. So I still want to plot by point, and then X and Y axis labels stays the same. I just copy and paste the rest. So the only difference is like we change the into our merged data frame of our UMAP results, and then we can give it additional information based on the things like sample type. When we run this, we will see our results being the shape of the UMAP is the same because the calculation is the same. It's just when we plot this, we label it by different colors based on different sample type. Does everyone get this result? Cool. And this is useful not only for just differ the sample by sample type, but because they have 100-ish different columns, they have a lot of like clinical informations we merged together as well. So actually we can try to play with some other criteria, such as we can merge it by different one thing is called like gender, for example. We can just copy and paste the plotting function, and then we just differ our results by different gender from the clinical data. And you'll see we have different criteria. If for some of the cases, if you're for some of your cancer cohorts, they have bias of that transcriptome based on this clinical information, you make some brand new discovery then. And also, yes, and that's because their clinical data set, that sample is not recorded by that. It's in their TCGA data. They didn't have that information for that patient. So that's why we have a non-recorded, something like that. And all of those clinical informations we can play around. We can check which of the information we can use. So we can use this column names function from our UMAP merge the data frame. It'll print out the entire potential doable columns that we can use as categorizing standards. For example, probably not all the samples have this pathology report, but some selected cases they will have. But all of them should have something like days to birth, days and years of birth, and their survival days. And we probably can, based on this, to categorize them as well. And the other useful information such as tumor grid, etc. So So why don't we try different criteria to the different UMAP projection to see how the other parameters looks like. And let's practice for 5 minutes with different criteria you want to cluster your samples. After that, we'll take a 10 minutes break. This will be the first demonstration of the downstream analysis. Let's start back at 2:55. And then we will go to the other example. So you probably have wondered, since we plotted this UMAP, it looks better than the PCA. Why do we plot PCA in a very early place before we generate that? And here is a potential reason that we need to be cautious when we use this UMAP on bulk RNA-seq, is we need to be aware of this potential batch effect. This is a simple demonstration which when I plotted over 33 different types of cancer from not only the project from TCGA, but also others, I will focus on issues like for the same control sample in the same organ, such as from the CP-TAC project lung control sample and the TCGA's project lung control sample, they cluster differently. This is typically shouldn't happen because they are all healthy lung samples. Their transcriptome should be quite similar to each other. There should be a unique cluster. This has happened because the so-called batch effect, which means like there could be multiple reasons, such as different technicians do this the experiment at different time. It could generate with different kits, with different version, or their normalization standards are different, etc. Those are informations. It's sometimes it's hard to get rid of this batch effect. So every time we run this UMAP, we probably need an additional function to check if our UMAP results is get rid of this batch effect. So the PCA plotting will be a good illustration. So for example, if all the control samples in the PCA and the UMAP are all clustered together, then the difference between different cancers can be considered as potential the real biological difference, because our control samples, they're doing it at the same time, they're okay, right? That's a good indicator. And another suggestion is like of mine is try to use the same projects like from same platform, such as use only from TCGA platform to compare different clusters when doing this UMAP, because their standards are unified by their own criteria, probably not from others. And that's my suggestion when you use this UMAP for clustering your own samples. Yes, so It is known like this. For example, our control sample, their transcriptome should be similar, and then they should be clustered together on TCA. And this is a good indicator to show, like, oh, okay, their controls are normal, they cluster each other, they're fine, which means that there's no significant batch effect in this data sets. Otherwise, if the control samples are scattered everywhere, and there could be a potential indicator that their data quality is not that good. So it could be so-called batch effect from different samples, collections, or data treatment, or etc. And then in this case, even though the UMAP are uniquely clustered each other, it could be mean something else, or they cluster into unique subclusters, it could be mean something else. So I always use another method to prove this UMAP is a good way. Basically, both of their structures will be the same meaning. It's just like UMAP have higher resolution compared to the PCA for clustering different samples. Yes. Yes.
What does the clustering look like if we grab these?
Uh, I didn't specifically shown here, but it looks like similar like this. When I'm doing the research, I used the two projects from TCGA lung adenocarcinoma and the TCGA LUST, which is a lung squamous cell carcinoma, which are both two types of cancer from the same lung tissue, and there are separate a batch they're different product doing in a different time, but all of them, all of their like 500-ish cases have about 100 control samples. I plot them together on UMAP, plot them together on PCA. Both of those control samples from different projects, they clustered together. This is a good indicator that, okay, different batch of the sample the control sample behave normal. Their transcriptome are similar between different batch, but different technicians. So, but they're actually their cancer samples in lungs, adenocarcinoma versus squamous cell carcinoma. They clustered separately into two different cancer clusters, just like in this published papers. They also use this UMAP to clusters. I think the blue one is squamous cell carcinoma versus the adenocarcinoma on the red. So, but the control sample they're clustered together. So it's just like or clarification that this data sets of the differences caused by actual transcriptomic differences. It's not due to the different technicians issue.
It's able to pinpoint in more detail, so sometimes it makes it so sensitive.
Yes.
It moves us differently based on.
It could be if the detail to if for example if we change that nearest the neighbor, that parameter too small, that we focus on too finer scale, we can still see some batch effect from others, and then it's always a good indicator that we've shown that control samples are clustered together as a or negative control, for example, because the control should be always the same. If like the control sample, their transcriptomics are already different, that means like biologically there are some issues. It's not, yeah.
I guess I don't understand the math of
Yeah.
so I don't understand why sometimes we break cells into different clusters.
It basically there's the mathematical formula behind this is too complicated to but there's that YouTube video, you can check that later on. There basically using some metaphor to explain it to you what are the difference. But even though the PCA have lower resolution compared to the UMAP, but for the very simple QC or quality control checking, and their control sample should always cluster together as the same trend as the UMAP show. If you saw the significant differences of the control sample clustering versus UMAP TCA or UMAP, that means that there's some potential issues of there's data structures. So probably that's the time you should have double check the quality of that data sets or just use filter out the outlier or something like that. Okay. So if there's no more question, let's go to the next example of this is the last example of this workshop, which is the survival analysis by Kaplan-Meier curve. I shown here is a demonstration of Kaplan-Meier curve. Basically, it shows the survival rate on a certain time point, which the x-axis is the time of our data collection, and y-axis is the percentage of the survival rate. And there's two concepts that we probably need to pay attention to when we draw this Kaplan-Meier curve. One is this tick. You probably have noticed before, like in the Kaplan-Meier curve, sometimes they have a little tick, even though the plot it's a flat line during that. That means like the sample is censoring, which the censor means like this patient's sample is it's not collecting the data after this time point for short. Another is the step. The step usually means like the patient is occurred with an event. So that two concept, event and censored. Event usually indicates that in our experimental criteria, in our case, it's life or death. If a patient is dead, they occurred an event called deceased or dead. And censored in the survival analysis usually refers to incomplete collecting of the data sets during this experimental period. So it could be caused by the multiple reasons, or such as the patient's clinical report stopped in the middle of the experiment, or after the certain followed after the followed by the entire collecting data point that patient still survived, which means that event didn't happen. So what we are going to do is based on this two, and then we will retrieve that clinical data sets from the TCGA READ project. We will manually define our event, life or death, with a deceased condition. And then we will manually categorize our patient sample into two categories based on one of our gene of interest survival, gene expression intensity. We will differ that our patients into two groups by the oncogene called CDK and N1A. If the one group, their expression level is, if the patient's expression level of that gene is above that level of that, we categorize into group 1, otherwise it's group 2, and we will draw a Kaplan-Meier curve for the survival rate, compare those two different groups on the selected oncogene expression intensity. Does this make sense? Okay. So there's actually two ways to retrieve the clinical data. For some of you, for the first case, when we perform that UMAP labeling, there's already some of the clinical information under that variant stable transformation or the RNA objects. When we load the RNA object, there's already some of the clinical information. But the more uniquely clinical data retrieve way is through a specific function called a GDC query clinic. So to retrieve the genomic data, we use the GDC query, and there's a specific function called a GDC query clinic that is where we retrieve all the clinical data sets. And we focus on a project TCGA read. And then let's nominate it into a new object called clinical query. Unlike the other functions that GDC query, which is into a different object, this clinical query will directly output our results into this table format that is human readable. And one thing is probably because this is just information from patient survivors, not genomic data sets. And here we have a total of 141 columns of all kinds of clinical data sets that are recorded in this project. But their column sequences for different products is different. So this is make us a little bit difficult for us to further extract a column. So before we do the analysis, we need to organize this table or the data frame to make it more neat and shows only the information we want to show on our Kaplan-Meier curve. So for plotting the Kaplan-Meier curve for the clinical data sets, we need three variables. One is called our patient's vital status. Another one is days to last follow-up. This days will be the information for those patients who survived. So we want to know the last day of their data collection. For the patients who are dead, we need the information of the days to death. Those three informations are typically recorded in majority of their TCGA project with this exactly the same term of their columns on clinical analysis. But their column orders might differ from projects to projects. So the first thing I want to check is those columns are all existing in our clinical query tables. So I am going to use a command called all, and to check are all of those three vectors as names of our columns are in our column names of our clinical query. Okay. This is to check our clinical information are all existing in the from this TCGA read project. and it report output as true, which means like all of those three columns are in that 107-ish columns of the data format. Now we want to know which is the column order are these three columns that we need that information. So we will change the command line from all to which. And then the logic needs to be revert a little bit. We want to ask which of those column names in our clinical query data frame are in our three labeled names. So now it outputs the column numbers that are three columns of which actually are. It's column 9, 107, and 113. So we can take a view of that three columns, whether they're actually the data by using the view command line. Inside that, we specifically annotate our clinical query data frame with that three columns. 9, 107, and 113. Okay, now that three data frame is days to follow-up, vital status, and days to death, which is the correct three columns, which is good. And then another thing we want to check is are all of the patients' vital status information are just live or dead. Like we checked earlier, there's some patient, their vital status information is unsure. So we want to get rid of that information because we need to make sure that the patient is live with that. We can use a function called table and then check the clinical query of our vital status column. Now it shows that the overall information, like 142 patients are live, 28 patients are dead, and there's one patient, their information is not reported. So we want to get rid of that. And we will set up a filter with our existing clinical query with the clinical queries data frame their information of our vital status. It cannot be equals to not reported. And we want to extract those rows whose vital status information is not this not reported. So after we filter this one, and then we will run this table, the command line to check again, we will see only, we will record only live and dead patient's information. Because when we categorize the Kaplan marker, each person's information needs to be either they're alive or dead. Otherwise, we don't know whether we should have a recorder.
And it's not the patient that is.
Not get, okay. This one, right?
Yeah.
So let me Typically, maybe due to a typo or separation. You can try to copy and paste that line of code. Okay, sure. You'll get this step right? No? Which you don't know how to check the ability to report it?
Yeah.
We just run that table command line again, table of our clinical queries, vital status column. Okay, that indicate this line of command is not successfully run. Typically, it's due to a type error. You can try to copy this line of code from the learning material that I sent you and then try to run that again. Something like maybe it's a square bracket instead of a curly bracket, like that.
And it's a case sensitive situation.
Okay, I hope everyone has filtered this out, right? Okay. And then to further use the package called survival and serve minor, we need to create a new object. Let's define it as diseased. So we will define a new column in our clinical query data frame called deceased, DECASED. And I am going to make a if else statement. So if our clinical queries vital status is alive, which means our patient is not deceased, then the selection will be false. Otherwise, it's true, which means the patient is still alive. And I can also check this deceased column information by the table of our clinical queries deceased column. Now I got 142 as false and 28 as true. So by this deceased column, which is a if else statement, like a true or false statement, we can define the overall survival date of our existing patient. So what we are going to do is we will create a clinical queries new column called overall survival make this column equals to a if else statement. So based on the deceased information. So if the clinical query's deceased column is false, which means the patient is still alive, then their survival dates will be clinical queries, the column from base to last follow-up. Else, the patient is not alive, their overall survival date will be clinical query data frames days to death. This part of code, yes. It will generate a new column of output like here. When we check the clinical queries output with our very last data frame, let's go to the very end. It'll generate the overall survival date column, which is we need the column with actual patient survival date and their survival status. Those two information will be need to generate that Kaplan-Meier curve. So by doing this step, we refined that clinical data formats with actually information we need. The next step will be integrate with our transcription data sets. And if you notice that our transcription data sets still in the ensemble ID, we still need to transfer the ensemble ID into the actual gene ID. Like we did yesterday, we didn't do that for today. So if we want to pick up a selected gene, we probably want to pick up a gene name. So we still need to do that ensemble gene ID transformation. So we are going to do the same thing as we did yesterday. Create that object gene metadata, make it equals to the data frame as data frame of our RNase row data. Which in this object, we have gene ID corresponding to the gene name. We will merge those two together to generate the information. Now, there's actually two ways to use the normalized count. One way is we use that variance stable transformation. Another way is like a DSeq output has a normalized count object we can use. So I'm going to show you another way. The normalized count is actually in the count function of our DDS object, which is our DSeq output. And then we give it a specific command line like normalized normal or normalized equals to true, which is we chose the normalized counts. Let's define this as the norm counts. Okay. when we take a look at the norm counts, it's actually normalized counts for actually gene counts. It's not a very unstable transformation way. This reflects by the different sequencing depths, etc., but it keeps the outlier information rather than the VSD transformation. So I'm going to use a function called this one. This one is a pipeline symbol that we can keep build up functions after this one. So I'm going to transfer this output into a data frame. And I am going to keep running by transfer the row names of this data frame into a column nominated as gene ID. I'm going to use a function called row names to columns. And then our new column's name will be as gene underline ID. Okay. So by running this is like we run these 3 lines of code together. So we first doing the counts and then transfer that output as a data frame and then redefined that row name into a new column as gene ID. So as you all see here, the row name is being transferred into this gene ID. This makes sense, right? The counts function extracts the actual gene counts from this DDS object. DDS objects is the calculated differential expression analysis, where their results should be like log2 fold chain of each individual gene, right? But they also have some intermediate output because each gene's normalized counts. Instead of the raw count that we take in the input, DSeq has a good way for normalize those raw counts based on their sequencing depth, based on the gene length, etc., other criteria. They'll calculate and normalize the counts. And we can use this function and give it a specific argument called normalize equals to true to extract those normalized gene counts.
So is the counts function under
Yes. If the memory was correct, or it's called a binary biogenerics, which is a dependent package of the seq2. Yes. Basically, they take all of their samples together, normalize sequencing depth, normalize gene counts, and they're using a very complicated Gaussian-like formula for the normalization. And you can check their manual page. There's a very specific description, but basically this normalization way in the DSeq is widely accepted by the peers. It's similar to RPKM, but they claim it's better than normalized with RPKM. Yeah.
this like normalized data is
Yes, we yes.
multiple occasions.
Yes. For example, if a sample sequenced 20 million reads for their sequencing depth, and the other sequence 5 million reads, and if you want to compare different gene expression level, that different sequencing depths could be a bias. That's why we use the normalized accounts. So after we do this transformation, we got the data structure from 444,000 rows with 177 columns. And we can check the data structure by another function called dim, and then give our row count, sorry, give our norm counts. And then it shows how many rows and how many columns our data frame has. What we are going to do next is a little bit complicated. We just need to transform the data structure from only a three column one, like a patient, each patient's gene, and then survival status, and then the overall survival date. We'll transfer this into a three column with mega lines of the data structure. And we can keep this with this symbol. And I'm going to use another function called gather. And then gather the key is our case ID. And then value equals to our counts. And then I'm going to remove that gene ID column. Okay. Now by doing this, when I check the data structure of our norm counts, it turns to a 7.9 million lines of and with only three columns. When we had to check the structure of that, it's like this. We have only three columns: gene ID, our case ID, and our counts. So just a transfer from a very square data frame into a very thin like data frame. Each line is, this case ID is duplicated, each case duplicated maybe 40,000 times. So that we can, we only did this to just to change the structure of the data for further analysis. And then we gathered together those columns by, there's a key argument, like which column do you want to gather from? Gather from this case ID, and then the value is from counts, and then remove an additional gene ID column. And this is when I do this, when I only remove that additional, this is minus is a remove an additional column. If I don't give that, it'll pops out something like without a successful transformation. And this is when I figure out when I debug it, when I use this one, it becomes three columns. So because if we don't remove additional, the gene ID column is duplicated and it generates an error. Yes. Gather command doesn't work. It probably takes some time. It's already finished. If you check on your console back, yeah. We only did this to transfer the data structure for the next step of the data analysis. And our next step is merge this from the gene ID with the gene names from the gene metadata. So what we're going to do is use this symbol to keep running command lines with our left join. And then the left data frame, I'm going to short indicate it as this dot. This dot indicates the data frame that we calculated from above. And then the second data frame is our gene metadata. And we merge those two data frames by the column gene ID. So after we're doing this, we will take a look at our data column. We have 7.9 million lines with 12 columns, which also has the gene names in this column. Now we've finally finished organizing the data structure. We can pick up a gene of interest that we want. We successfully merged the clinical data sets with normalized transcriptomic data sets, and we transformed the gene ID into the gene name. Now we can pick our gene of interest. I'm going to define a new object called short for GOI, short for gene of interest. And it can be extracted from our normal counts columns. And I'm going to use the additional column function called filter, which is filter the information from that data frame. And the gene name, I'm going to filter the gene name equals to any of our interest genes. I'm going to use the gene CDKN1A as an example. Now, when we take a look at the gene of interest data frame, we only have 176 entries for 176 transcriptomic data sets and 12 columns for those. We have the normalized accounts, and we have the gene names are all CDKN1A, and we have the barcode for each individual patient cases. But we need to, we want to further filter this out because there have some additional information from control patient samples, transcriptomic data sets. If we want to compare the patient's survival, we probably want that patient's transcriptomics from their cancer samples, not their normal samples. So each case indicates a patient. Some of them has 2 transcriptomic data sets recorded here. One is from their cancer sample, transcriptomic intensity. Another one is their normal samples, transcription intensity. So we want to compare that person's actual cancerous tissues transcription level, right? So that's why we introduced the TCGA barcode system in day 1, because when we pay attention to the case ID barcode, there's 2 kinds of the barcode. One is focused on the 01A. If you remember the content from day 1, from 01 to 09 indicates a patient's a cancer tissue. And from 10 to 19, such as the example on this 11A, indicates a healthy peripheral normal tissue. We want to get rid of this normal tissue. So our next step is filter out transcription data from that solid tissue normals. What I'm going to do is create indices called tumor indices. Make it equals to grab a function. I'm going to use this function grab from whatever the barcode, as long as there is a short for -01A dot star, which indicates anything within and after this. And from the gene of interest case ID column, I will record this as our tumor samples indices. And then when we type this tumor indices and take a look, it reflects the number of our cases, which are actually our tumor samples. So we can, based on this index to filter our tumors gene of interest. Make it equals to our gene of interest data frames tumor indexes. Now, this tumor gene of interest data frame only contains 163 samples for all of them are cancer patients. Now I am going to use an example to separate our tumor group into two based on their median level of their transcription. I'm going to create an object called median gene of interest, and then do a median function to calculate the tumor gene of interest data frames counts. And check that number. It's 3,800-ish. This is the median normalized count from our group. And then we can define a new column in our data frame from our tumors gene of interest. Let's define it as a strata. Make it equals to our if-else statement. So if our tumor groups counts are above or equals to our median level of our gene of interest, then we define their status as high, which is their high expression group, otherwise is low. Yes. Make this a factor. I don't think it's a factor, right? You check that. No, we don't have to. We don't have to. Okay. We can check our re newer structurally sure defined as use that table function to check how many are defined by each other. So since we define subcategorize them by a median, they should be almost equally distributed. But this function, you can further use your own criteria, such as if you want to subcategorize them into 4 or 5 based on your criteria or the group of genes of each expression level or even mutational data sets, you can use that as well to manually define this. So now our table is transcriptomic data sets with counts, with gene names, with subcategorized based on their gene of interest expression level. And the final step is merge our clinical data with this data frame. And our clinical data has a column for submitter ID. If I show you here, it should be looks like this. I'm going to use a head and then a clinical query data submitter ID. They're shown this with TCGA program name, project name, and then the patient's ID without the additional sample name, etc. So to merge those data frame with our tumor gene of interest with actually the very long TCGA barcode, we need to delete the additional information after this sample ID. So what we are going to do is I am going to define that new column for the trend barcode one as a submitter ID column. Submitter ID. And I'm going to use a function called gsub, which is to grab and replace that column that has and after has the label of 01A with and after that one. And I'm going to replace it with a single quote inside of them is empty, which I replaced everything with this and after that into an empty one to train that off from our tumor gene of interest data frames case ID column. So what this function will do is it'll generate a new column called submitter ID, which I replaced everything after and with this 01A into that. So this submitter ID is corresponding to the clinical data frame that we can use this information to merge them together. So I'm going to merge our tumors gene of interest data frame with the function called merge and then our tumors gene of interest with our clinical query by the column called submitter ID. And now we finally merged the clinical information with all kinds of sorts for the gene of interest that we picked up for this. Now we have 156 of the clinical information with the finally with overall survival date and the disease column that we created earlier. So after this, we can use the survival and the serve fit from the survival package to generate that survival curve. I'm going to create an object called fit, make it equals to serve fit. On this overall survival, and then our condition is deceased. And then subcategorized by our sample, this is the same one as our subcategorized DC condition, subcategorized by the strata column. And then the data of input is from our gene of interest, humerus gene of interest. What this function generates is the overall survival calculation of this fitting data, how many in the median survival date, etc. And then we can finally plot this through a function in their survival package called the ggserv plot. And then fit equals to our fit, data equals to our tumor's gene of interest, and p-value equals to true. And then we also want to print that risk table. Yes. Oh, that is because the overall survival from here, when we organize the clinical data sets, here. Maybe this line of the scripts, you have a type error or something. Make sure that the overall survival is exactly the term with no like a case error or something. If this is okay, then is this overall survival input, this has some type error. Okay. Have you all generated this figure? Okay. Yes. Here. I just called a fit of all the results. It default shows a summary of this information. Sure. And this is the final plot or the examples I'm going to show you for today. I'm going to enlarge it a little bit. So this is shown in two groups of our gene of interest, CDKN1A expression level. And the p-value shows the log rank test between our two groups, whether it's significant or not. And then at the bottom, we can see the number of risk, which is in a certain time of point. In our case, how many samples are still, how many patients are still surviving at the time point. And that's the parameters for that risk table plot for true means. So this is just a simple expression that we can differ that by a single gene's expression level. And in the future, you can merge this with your like DNA methylation data sets or other things you want to and create your individual survival plot of your interests. So at the end of this workshop, I want to thank you all for the participation, and you're all awesome to run through all this complicated code. This is actually my first time of generate for this workshop, and I'm glad that all of your codes are, or most of your codes are working. And I really appreciate your suggestion on this feedback to give me suggestions so that we can further improve this workshop's quality. Thank you all for the participation, and good luck with your research in the future. Thank you.